everyone's guts are going to be different, whether it's a human or a pet. We're all exposed to different things like different households and different environments, or maybe perhaps you or I eat a different diet, or perhaps two different dogs can eat different kinds of diets as well. There's a lot of external factors that can influence the microbiome.
Well, hello, welcome to the Pet Food Science Podcast. I'm very pleased to be here with Dr. Martini today. We're going to talk about gut health and maybe how to optimize nutrition to maximize gut health. I think we'll have a very interesting conversation. Dr. Martini, if you would, please just tell us a little bit about yourself and how you got to be in the position you're in today.
Yeah, absolutely. First of all, thank you so much for having me. I'm definitely excited to be back and kind of talking about some of the things that I'm pretty passionate about. A little bit about me is that I just recently completed my PhD program here at the University of Illinois, all in animal sciences and specifically companion animal nutrition. I did do my undergrad and my master's here as well, kind of within the same realm and within the same advisor. A lot of my research really focused on the microbiome and trying to use nutrition to help with recovery, especially after antibiotics, just because one, they're really bad, but two, they're also widely used in veterinary medicine. And so a lot of my research was trying to focus on how can we improve those recovery parameters, or is there certain things that we can do to help with benefiting or improving pet health? A little bit before that, I am actually originally from Connecticut. That's where all of my family's at. But I also grew up as a Navy brat, if you will. So I kind of lived a little bit all over the East Coast and had a great time with it. And now here I am.
Kemin Nutrissurance is compelled to provide safe and delicious ingredients to improve our pets' food so they can have the best quality of life. Every day, they deliver specialized expertise, innovative products, and unrivaled support through the pet food and rendering value chain. From oxidation control and food safety to palatability and nutrition, all the way through a suite of tailored services that support you from start to finish, we ensure you're getting the most from Kemin ingredients. Kemin is compelled by curiosity to ensure pets around the world are living happy and healthy lives.
Well, thank you. That kind of gets us up to where you are. I wonder if we could start out by just, you stated, well, antibiotics are bad. And I think you meant bad in the sense that they really disrupt the gut. They may be very helpful to the pet, right? They may be the right thing to do. Then the less than desired consequences are what's left. And so how do you, let's ask and discuss how we decide what a healthy gut is. Where are the measurements and how do we know what that is? What do you think?
Yeah, so you are right. Me saying that antibiotics are bad is definitely very individualized, but they can be really good in certain circumstances. Like if an animal presents and they've got a huge pathogenic overload or bacterial overgrowth or something, antibiotics can really go in and disrupt and maybe help that animal. But there's a lot of negative side effects or consequences, if you will, like loose stools, like pet owners don't want to see those kinds of things, can kind of upset the gastrointestinal tract. They may not want to eat. They may not want to, kind of be the normal selves. That's what I mean by antibiotics are more so bad. But whenever we're really thinking about what is a good gut, what is a great gut, if you will, we're looking at kind of like a complete balance, if you will, of the microbiome. We're looking that not only it's functioning in its own little microenvironment, but it's also doing things that benefit the host. And so kind of to rephrase or kind of redefining it, we're looking for good function. We're looking for good output. We're looking for things that otherwise aren't negative side effects. Like we're not seeing things like loose stool or anorexia or weight loss, for example, that you can see a lot whenever that microbiome is disturbed. So it's really kind of looking at that there's no complications, that there's no issues that are kind of arising within an unhealthy gut, if you will.
Oh, that makes sense. I think that's, people want that healthy gut, and they certainly observe it with the things you're talking about, like good quality stools and healthy appetite, those things. One of the things that sort of interested me is this whole capacity for understanding has increased, and it's just marvelous, right? We do stuff that when I started my career, governments couldn't afford to do, and now lots of folks are doing them as far as measuring what's there and maybe how that's influencing the gut. But seems like a healthy pet or a healthy, human can be quite satisfied with really quite a different set of microbiota. Is that your opinion? And how does that relate to if I go in and measure a gut? How do I know what healthy is?
Yeah, everyone's guts are going to be different, whether it's a human or a pet. We're all exposed to different things like different households and different environments, or maybe perhaps you or I eat a different diet, or perhaps two different dogs can eat different kinds of diets as well. There's a lot of external factors that can influence the microbiome. But really, ways that you can kind of quantify it or getting down into like the nitty gritty details, if you will, you can run different panels that you can measure different bacteria. So for example, Texas A&M does a qPCR panel for veterinary clinics as diagnostics, and they can really kind of fine tune and look in on different bacterial species that may or may not be present in an ill patient compared to a healthy one. So you can really kind of quantify those on a very, very small scale. But what you see visually is going to be things like if your animal is vomiting a little bit more, or perhaps they're losing a little bit of weight, or their stool quality gets kind of bad. Those are little bits of ways that you can kind of monitor the gut health in that realm. Even if it's just a visual description, even if you're not like in my home, I'm not going to be sequencing bacteria, but like if I'm starting to see that my animal maybe has some kind of imbalance that's showing up in a looser stool, those are things in which can be monitored. If you start noticing like increased frequency or like I mentioned, stool quality, there's some more visual characteristics that you can kind of hone in on as a pet owner, or even kind of just as, us as people, us as humans. We notice these kinds of things just within ourselves. But those are really important ways in which it can be monitored as we start seeing changes like that.
Yeah, I guess I'm still that makes all sorts of sense. It's what we observe, right? That's what we can find, and you don't have to walk into the lab to understand that. If you have a pet with loose stools, that's just really, really not what you want to have. But I'm trying to understand it. I mean, how is it that interaction of maybe it's the genotype or the dietary or the pet that seemingly very different microbiota can end up with really pretty healthy pets. In some ways, the disease becomes more similar, right? If you see a bunch of Heliobacter jumping through the microbiota, then you're going, this is sickness. I recognize that. But seems like healthy can be so diverse, and it's actually surprised me once I learned that. Is that how you feel, or is that what you're seeing?
Yeah, I think healthy is a very broad term to use. Kind of like I mentioned before, it's very individualized based on different people. But to me, healthy always kind of meant, an absence of issues or an absence of ailments or an absence of certain perhaps pathogens. But that to me was just an amazing concept, because when I first started, I thought, oh, healthy is healthy. There's no way that they can have multiple definitions. But then really a lot of my personal challenge was during my PhD. It's okay, I've been saying it this way, but how do we define it? And even then, the definition became very broad scale as well, but it became more about resiliency and resistance to infections, perhaps, or really kind of like really honing in on the absence of issues. But it became very broad. So you do make a good point.
Yeah, it's fascinating. Well, I know a number of people, I'm not familiar if you've written about this or not, but they really talk about this dysbiosis index. I think that's got some value, but I do wonder, because you can make a pet become more diverse by adding a pathogen, and that's not if you look at diversity and homogeneity within a grouping. How do you feel about that? Is it a good tool to sort of know what that index is?
I think dysbiosis, like the index itself, can actually be very effective in clinical applications. So looking at a patient with disease, simply because the dysbiosis panel is really built on core bacteria, but also bacteria that are known to cause GI upset. And so if you get an animal that presents in the clinic, you can easily measure and see, okay, they've got an increase in like E. coli or streptococcus, or, reductions in some of the good core bacteria, if you will, like fecal or Peptacetobacter hierinonis. But really, in healthy animals, the dysbiosis panel, you don't expect it to shift. Like, you don't want to see extreme dysbiosis, but it can be helpful for getting things like baseline values. If you have like a young animal, you take them in, okay, this is where they start And over the years, you can kind of monitor and see if, you're starting to see trends or things like that. But even more recently, some of the research that's either come out of our lab or companion labs has really been finding that dysbiosis can be affected by different diet types. And so, like, raw food will change the microbiome of a pet compared to a pet that's eating a kibble diet. And so you may start seeing shifts or perhaps increases in dysbiosis simply due to diet. And so it's kind of just really important to understand that dysbiosis isn't always necessarily a bad thing, but you can use it to look at clinical patients. And you're starting to see, if dysbiosis is increasing, it's probably not the greatest sign in the world. So you want to figure out a way to tone it back. But I think to answer, I think it can be really effective, especially in clinical settings.
Well, that makes sense. Well, you talked about this microbiota and we have the capacity to monitor it. Do you think that, we could gain by having some, longitudinal, through the life of a pet, a microbiota analysis and say, like you might look at blood urea nitrogen over time or cholesterol or some other markers. Should we be thinking about doing that with microbiota as well?
I think it could be really interesting to do. I mean, personally, out of my own interest, just because, this is the field I've been working in, I think it would be really interesting to look at where my dog first started versus where she ends up 10 years later. But I think also, too, what we're finding a lot in human medicine is that perhaps there's some predictors and shifts in the microbiome earlier in life that are leading to things later, like colorectal cancer, or perhaps IBD and little predictive models like that. I do think that a longitudinal study would help with possibly finding either new biomarkers or little shifts and nuances that are starting to show up that could be leading to secondary things. Realistically, I think right now, in vet clinics, I say we, but veterinarians are really kind of screening for parasites in the fecal test, not necessarily getting that longitudinal data. But I think it could be interesting as like a little addition, if you will, to kind of see, like I said, where they're at and versus where they end up in years from now.
Well, let's talk about that. I mean, if I wanted to recommend a fecal sample, could you do that by sampling? I used to call like a little loop where you'd actually go in and grab some fecal sample right in the pet, or do you need a whole, large, 200 grams or whatever fecal sample for an analysis? Is it one way better than the other?
In my opinion, and really kind of my experience, when collecting fecal samples just within our lab, we're trying to get to the center of a fecal sample just because that's the area where there's like the least amount of oxygen. And it's kind of a little more preservation, if you will, of what it was like within inside the host before it was excreted. And I mean, again, I may be an over-the-top pet owner just because this is the realm that I work. But when I collect samples to submit to my veterinarian, I'm collecting the whole samples so that they can go in and take the middle piece out, like trying to keep it as preserved as possible once it gets to that test. Just because things like oxygen or, perhaps my grass has a fertilizer on it, that can really affect some of the measurements that you can get from a fecal sample.
That's interesting. Well, what do you think? I mean, if you look out the future, is there better ways of doing this? Has it become, sort of within the realm of a veterinary clinic to think they could be doing this monitoring over time? Or how does it look in the future, do you think?
I think with more research that's been coming out, especially kind of in my discussions with different vets or vet clinics, there's more interest in trying to do some of these measurements earlier on, but where a lot of the resistance comes from is sometimes the pet owner perception. I love my pet, but perhaps the perception could be, well, I don't want my animal to become like a research subject, or I don't want to volunteer them and things like that. But I think more veterinarians are kind of shifting towards the more perspective and longitudinal approaches, where if they get a new patient in, let's get as much as we can and then watch it as it goes. I think it could be something that the industry might be shifting towards, but just with anything else, I think it's going to take time and a little bit more acceptance and understanding from the pet owner's perspective that the data doesn't lie. Like, research isn't going to lie over the years. I think it's really important to also increase education too. Like, I'm not going to be screening for things because I know something's there. I'm going to be screening because I want to make sure that nothing will be there or show up later in the future. That's really kind of more on the approach that I would like to hope that clinics or perhaps that world can go to. I would love to see a lot more pre-issue collections, if you will, before anything shows up, on more of that preventative approach.
Yeah. Well, what do you think? We've talked a lot about understanding the microbiota and how, actually maybe we could dive that way a little bit. Microbiota has a pretty large influence on the pet. And maybe we could talk about the biotics that are coming out of the microbiota and maybe some of that crosstalk that's going on. How does it influence the pet? What happens in the gut that matters to the pet?
Yeah, so the microbes are super important because anything that a host doesn't digest, for example, fiber is one of like our favorite topics in this lab. Anything that passes the host digestion goes to the microbes, and the microbes can take it, they can turn it into things like energy, perhaps a protein source for growth and reproduction, but ultimately anything that microbe produces, the host can then absorb. And so one thing that my research really kind of honed in on was bile acids, just because those are one of the main products that bacteria can produce and then the host can then get. And so really what's important is that you don't want microbes to be overproducing certain things or perhaps increasing production of something that's going to harm the host. What we like to measure and what we really look at as good things are things like short chain fatty acids, just because those we know are going to be good energy sources, whether it's for microbes or for us as a host, for our cells and things like that.
Well, let's talk about those bile acids. That intrigues me. So these are things that are coming from the host. They're being metabolized by the microbiota and they're being reabsorbed by the host. I mean, it's a perfect opportunity for sort of the host to curate a little bit of the microbiota, or maybe even to understand, I always put an intellectual thing but to understand what's going on in the gut. Is that what works, or how do you see that happening?
Yeah, so bile acids originally start in the host. And so they're this primary form, if you will, but it's ultimately the microbes that convert it from one form into another. And for the most part, that secondary or really that byproduct is what's reabsorbed in the host. It's not a perfect system though, just because some still ends up in the sample or in the fecal sample and it's excreted, so it's lost. But we can kind of quantify that and see what's coming out, because if there's an overproduction or an underproduction, we can quantify and say, maybe something's going wrong here. Maybe there's some dysfunction or dysmetabolism. But really, that's like what we can really kind of focus on or really quantify within a sample and see, like I mentioned, if there's something perhaps a little imbalanced.
Well, what would you measure that would know it was imbalanced?
We can measure both like the primary form and the secondary form, but more specifically within dogs and cats, there's only one genre and one species really of bacteria that can convert those, and it's called Pepticetobacter hirinonis, or it was just renamed from Clostridium. But really, that is the only bacteria within the host that is capable of converting primaries into secondaries. And so not just measuring those byproducts, but if we look at that actual microbe that's converting it, and let's say we see a reduction, we're going to see an increase in that precursor because that microbe's not there to convert it. And so if we can really quantify or look at those changes, we would very much early on know this is where this imbalance is coming from. So not just measuring the products, but then you can actually look at like that sole species that you know of that is either missing or it's not working the way that it's supposed to. That's what we can quantify. And it's really fascinating, in my opinion.
So it's not really, if the gut is sort of using the bile acids, or the host is using the bile acids to monitor the gut, it's really pretty much monitoring that one species, or is there a lot of interaction with that species and the other microbes in the gut?
Without diving into a biochemistry lesson, that microbe is really the only one that we can focus on or look at. There's a little bit of crosstalk because some of the microbes there actually perform a smaller step, if you will, right before it, where they can cleave off an amino acid or they can kind of change the form a little bit so that then Pepticetobacter can come in and then convert primaries into secondaries. And so there's a little bit of crosstalk there, really kind of cross interaction, if you will. But really, it's that one that's primarily doing the job. It does have to go through a smaller step first, but that first step is pretty much always being done just because there's always those microbes there that have that ability. It's just that one Pepticetobacter that if it's gone, it's not going to convert.
So does the pet respond to that? The amount of secondary bile acids that are suddenly available, is that something that it would respond to as far as trying to moderate what's going on in the gut?
As far as I know, it's not really something that like a pet owner can see. If there's bile acid dysmetabolism, where there's an over quantity or an over volume amount of like the primaries, right? If we see those quantified in the stool, absolutely, but that's measuring it within a lab, but you're not going to visually see it. And so some dogs that do have bile acid dysmetabolism will show other signs like anorexia, but it's not going to show up in something in the fecal sample. If your dog naturally pukes or if they just have a higher tendency to, you may not notice anything or may not attribute it to being bile acid dysmetabolism. That really requires more further or perhaps more invasive testing to look at it. But unfortunately, it's not something that we would see within a fecal sample that I go out of my backyard and pick up. Perhaps it'll be a little bit more loose, but I'm not going to know that's just bile acids. Perhaps I'll just think, it's a little mucusy today.
Well, what do you think about ways we might moderate the microbiota health? I mean, I think we've established, or people know that you'd like to have an optimum microbiota. What sort of nutrients might influence that and improve that?
So there's so many things that can really kind of modulate the microbes, but as a nutritionist, we're looking at more of the macro scale. We're looking at things like dietary protein, fats, fibers, carbohydrates. We can really get into the nitty-gritty there. But if you look at a diet that has lower fat or higher protein or higher fiber, all of those are ultimately going to influence the microbes because, like I mentioned before, whatever the host doesn't digest or if there's an excess of, the microbes are going to get to that and they're going to have a heyday with it. So really, as a nutritionist, we look at things that we can modify. If a dog is presenting with certain symptoms, perhaps like if they've got constipation, right? We're going to look at maybe a diet that's got more fiber to help with gut health. But it really comes down to maybe perhaps like what that dog is presenting with or what clinical signs that we're actually seeing within it. But even just besides that, even our nutrition trials, all of different diets all have different constituents or different compounds or percentages of things, but there's a lot of influence.
Well, let's dive a little bit deeper. One of the things that fascinated me, I sort of come from a space where you want to have optimum by not having too much of something and not having too little of it. And that optimum for protein was sort of, there's some differing opinions, but we saw a benefit a number of times for renal health, for having an optimum protein rather than maybe a maximal protein. And we suddenly found, over the last few years that it was these, I call them postbiotics. I think the microbiologists might want postbiotics all to be positive. But, the things that the microbes are producing that actually were toxic that I sort of never pulled together. I kind of thought, protein causes more glomerular filtration, and so maybe it's just a workload. But in fact, it is the microbiota, shifting to that proteolysis, and then these negative compounds are resulting from that. So that's one. Do you agree with that? Or, that's kind of me thinking aloud, but what do you think?
I would agree with that. I really do think that dietary protein, like you mentioned, it has a huge effect on the kidneys, and it's things that we can quantify looking at like blood samples. There's so much that you can get from a blood sample or a fecal sample, but really if there's too much dietary protein, like you said, it's going to switch to perhaps a more proteolytic environment or those kind of metabolites. And then you get high production of things like phenols and indols, which people think are terrible. We don't want to see those anywhere. And I fully understand it because they are a little sneaky. But really trying to, if there is an animal that does have kidney predisposition related issues or symptoms or signs, perhaps a lower dietary protein diet would be great, or a hydrolyzed protein. And like really looking at those kinds of nuances or those tiny details in a case can make a huge difference on not just the pet health, but the pet's well-being.
So certainly protein quality matters in there too, so that more of that protein is being absorbed and not readily digestible. Because the microbes, like you said, they take what's available, right? And if there's more of a particular type of nutrient, I guess, coming at them, then different microbes flourish. Well, let's talk about fiber, because that's a very complex issue, isn't it? So there's lots of, like, people talk about soluble and insoluble fiber. And how do those things relate to the microbiota?
Yeah, so kind of starting out in this lab, I had a very small picture on how important fiber was, but then with my advisor, both Dr. Kelly Swanson and Dr. George Fahey, both really got me on the fiber train. So to answer your question, we're always looking at different fibers because different fibers have different characteristics or different properties. Some have higher soluble fibers and some have higher insoluble fibers. But really, when it comes down to, like, for example, what I may want to use or as a supplement or perhaps in a certain dietary trial, I may look for those characteristics. Like if I'm looking to make a diet that's got, like if it's a wet food, for example, I may want a fiber that's got more soluble fibers in it simply because those can really bind up water and they can make things a little bit more gelatinous versus insoluble fiber may help with structure, but it's not really going to bind water the same way that we look at it. And so in kind of relating it to clinical patients, if a patient comes in with constipation, if I give them too much soluble fiber, they could have extremely looser stools. They can have high flatulence. And those are things we definitely want to avoid, especially within those patients.
Yes.
But in cases of things like constipation, those insoluble fibers are really that fraction, like cellulose, for example, can really help the pet owner with laxation, kind of promoting the movement of the GI tract just because our microbes can't process things like that. And so there's a whole bunch of nuances. And I love the world of fiber just because, like I said, not every fiber is going to be the same. They've always got little differences, or whether it's a structure or a different binding or different bond in certain areas, that makes a difference in how it really kind of reacts within a host, whether it's a human or an animal.
Well, it's not exactly fiber, but it certainly sort of acts like one. I'm thinking about this bypass carbohydrate that maybe is in a resistant position, and then it survives the initial digestion so that it's available for fermentation. How does that compare to these fibers you're talking about?
Yeah, so I think what you're talking about is resistant starch, where it totally, it can act like kind of like a prebiotic, if you will, or kind of more in that realm where in a host, it can be absolutely metabolized by a bacteria. But in dietary fibers, those are really more in plants versus resistant starch is kind of a carbohydrate found in things like potatoes. And so that's a huge difference between prebiotics and dietary fiber that I kind of want to make is that dietary fiber comes from plants, but prebiotics are not plant derived. You can find them, but they're not necessarily structural within a plant. And so that resistant starch that goes through like the processing, that can then, it's not going to be digested by the host. So it acts in a little similar way.
Well, I think you use prebiotics in a way that's very defined to the microbiota world. So can you help us understand what you mean when you say, hey, this is a prebiotic and what that means?
Yes. So prebiotics, according to ISAP, or the International Scientific Association of Pro and Prebiotics, they really define a prebiotic as something that is selectively used by microbes. And so not every single microbe that you come across may be able to handle certain compounds, but a prebiotic is designed to affect the microbes, or it's designed to really kind of target certain species that can help with short chain fatty acid production, for example, like the ones that we know that are very tried and true and great at, short chain fatty acid metabolism. Prebiotics can really target those to help with increasing it. So really that difference, or that key word, if you will, in prebiotic definition is selectively utilized, or I guess it's two words, but selectively utilized by certain microbes to have an impact.
So it does, like we used to sort of think, again, I'll show my age, that prebiotics were kind of things that resulted in enhanced bifidobacteria. If you did that, then you had a prebiotic. It must have been wonderful. But you're saying that it's really now a word that kind of defines a microbiota, always healthy, right? So something might be used by a microbiota that would turn the wrong way and be toxic. And so even though it's being used by microbiota, you wouldn't call that a prebiotic as you're using the term.
Yeah, the prebiotics that we really focus on, or that are kind of defined, are selectively utilized, but also have a known health benefit or have a positive outcome onto a host. And so the usual ones that we focus on are things like short chain fatty acid metabolism, just because we know that's a really great intended outcome. We love to see high short chains always in a sample. If you see low, then perhaps there's, like I kind of said before, a little dysmetabolism. But you want to make sure that whatever prebiotic you're giving is having a very positive health outcome.
So again, maybe people might not be so familiar with the term. So short chain fatty acid would be like acetate, propionate, butyrate. Are those only ones, or are there others that I should be thinking of?
Those are the main three that we really focus on. You can get even deeper and talk about branch chain fatty acids.
Yeah, I was going to talk about that. How do you feel about, so you can get some branch chain fatty acids perhaps that don't have the same benefit that good old butyrate has, right?
Yeah.
Let's tell, what do you think about that? I mean, is it because of different microbe, or is it a different source, or why would you have maybe this less than beneficial, or at least not as beneficial as maybe butyrate, which is, I like propionate, acetate, and butyrate, but maybe not isobutyrate quite so much. Or what do you think?
Yeah, so it's not that we're trying to using diet or using nutrients or anything like that. We're not trying to decrease branch chains, but we don't want to see an increase, because where they really come from is additional protein metabolism. And you can really get into the weeds on that, that biochem lesson there too. But what we're trying to make sure is that we're getting short chains, because those are the actual energy sources either being used by microbes or they're being used by host cells. But an overabundance really of those branch chains can actually become more harmful than good. And so usually what we're trying to look for, and at least in our nutrition studies, is we're trying to make sure that either the levels are staying the same or perhaps they're going down a little bit. But there's also other protein catabolites that you could really look at, like the phenols and indols and more of those volatile compounds. But branch chain fatty acids, as far as my research has always been concerned, we're just trying to keep them either the same, if not a little bit lower.
It's kind of a general thought, isn't it? That there's lots of things that we exist in biology very nicely at the right level. Everything probably, I mean, I can imagine having too many short chain fatty acids. I've run studies. I have stories. But you have to get really strange foods before you get worried about too much butyrate. But you want a balance of these things, and you want to maintain that optimum. And I guess that sort of spins to my next thought, which really is, what do you think the long-term effect, if you can optimize this, is on if you have a healthy pet that's staying healthy? We've sort of talked about the disease pet and trying to move them back towards health, but maybe what's the long-term effect of having this optimum microbiota that we're talking about?
Yeah, that's a really great question, just because, like you said, we talk a lot about, okay, we have a diseased animal, let's get them back to normal or healthy, if you will. But how do we keep them healthy, or how do we maintain that healthy microbiome? And really, if you're going to use the word optimal, or how I'm going to relate this back is how I define what healthy is. It's the ability to either resist or be resilient in certain conditions, like a pathogen issue or a bacterial issue. But really, a lot of this is on the preventative approach. If you have a very great high functioning microbiome, you're not having issues, or you're not feeling GI disturbance, really improving or really maintaining that health as well can help with preventing things like future infections, or in patients that have had disease and come back to healthy, you want to maintain that so there's no relapse in the future as well. And so really the preventative approach is helping with things like resiliency, or perhaps some resistance to some capacity if there were to be an issue.
Well, it sounds like there's a pretty big benefit for a pet to making this, as best we can as nutritionists.
Yeah, there really is, just because a pet can't talk. And so that's something that some pet owners, I mean, I myself struggle with it, but they can't communicate with us. So what we see, like in their stool or in their behavior, that's when we notice that there's something wrong. If we do what we can as pet owners to make sure that, nothing bad happens, we try to give them all the right things. That's what we can do as pet owners, if you like responsible, if you will, but making sure that we're doing what we can to help them so that nothing bad happens. Because I don't like to see my dog sick. No one likes to see their animal sick.
Sure.
So the more that we can kind of do on that preventative front, or making sure that we're doing what we can, is really important in my opinion.
Makes sense. Well, Sarah, thank you. I've enjoyed this component. I did have a couple of questions. You've just completed your doctorate degree. You've come out of a good lab where I'm sure you've had a lot of positive experiences. I wonder if you might just tell us a little bit about what you would see as maybe some of the things outside of your direct science that you learned in this, and maybe if you were looking for a team or wanting to lead a team, what you might sort of wish to have, a desire in that.
Yes. So throughout my PhD, a lot of my stuff was very teamwork heavy. And so the qualities are really those characteristics that I found to be as like successful team is all of us were really encouraging each other to stay curious or ask questions or come into situations with an open mind, just because you never know what's going to happen or what you're going to learn that day. And I think that really plays into the ability of a team success or a person's success, or even as a leader, you want people to ask questions. You don't want to, kind of make it so that nobody can. You don't want to make it uncomfortable, if you will, but being able to, keep an open mind and be curious and never lose passion. Those are the big things that I've kind of noticed either throughout my program or even in, my little side quests that have really made for an effective either team or even talking with like companies and sponsors and things like that.
Well, that sounds pretty exciting. Well, I've enjoyed our chat, and I think that I've learned quite a bit more about what this is about. I hope that others have seen it as well. So perhaps we'll sign off. Thank you for your time.
Thank you so much. I look forward to our future. Yes.
This is the Pet Food Science Podcast.